In this video, we explore the topic of screening. What does the term screening mean? In molecular biology, screening refers to various methods used to identify different phenotypes in a heterogeneous population. By screening or testing the cells, information about the functionality of the target gene can be obtained without affecting the growth of the cells. While traditional screening methods often have low throughput, they can also provide semi-quantitative information. Newer screening methods or modern laboratory equipment can significantly increase throughput. Classic examples of screening include the blue-white screening. This method uses an E. coli strain XL1 blue that has a deletion in the alpha peptide, a subunit of the beta-galactosidase, making the enzyme unable to hydrolyze galactosides like lactose or X-gal. X-gal is a lactose analog that produces a blue dye when cleaved. In classic blue-white screening, this strain is transformed with a plasmid carrying the gene for the functional alpha peptide, complementing the beta-galactosidase and enabling the cleavage of X-gal, resulting in blue colonies. When cloning with the plasmid, the target gene disrupts the alpha peptide gene, preventing the production of a functional beta-galactosidase, causing the colonies to remain white. Thus, the genotype of colonies can be distinguished based on the blue or the white phenotype. Another form of screening is enzyme-catalyzed screening methods, such as detecting pH changes using pH indicators. If the target gene encodes an enzyme that catalyzes a reaction that alters the pH, the enzyme can be detected. An example is pH change in a reaction catalyzed by an esterase. Depending on the equipment, cells can also be cultivated in 96-well microtiter plates, and enzymatic activity can be detected using a photochromatic assay. An example is the color change in the DNS assay, which is used in medicine, among other things, to detect the enzymatic activity of alpha amylase. The term screening should not be confused with selection. What does selection mean? Selection involves various molecular biology methods used to select different genotypes in a heterogeneous population. By selecting cells, different genotypes are distinguished by influencing cell growth or metabolism. As a result, some cells may gain a survival advantage, while others are eliminated from the previously heterogeneous population. Selection always has a high throughput and is often more cost-effective than screening. Antibiotic-resistant gene selection is one of the most common selection methods. When a heterogeneous population is cultured on agar plates containing antibiotics, only strains with antibiotic resistance will survive, while other strains are eliminated. Another example is the use of auxotrophic strains. Auxotrophic strains cannot synthesize essential components for cell metabolism. For instance, a strain unable to produce arginine must absorb it from the environment to survive. If arginine is unavailable, the cells will die. The mechanism can also be used for selection. In this course, we will perform a screening in the form of a filter activity test. This method is linked to the enzymatic activity of our target gene, with esterase as the example. The esterase gene is cloned into a PET vector system. Since the PET system exhibits basal expression even without induction, cells cultured on agar plates will already contain small amounts of esterase. The dye reagent in the staining solution serves as a substrate for the esterase, leading to a violet precipitate in positive clones. Colonies lacking the esterase gene will initially remain colorless, but slowly turn violet after a few minutes due to endogenous E. coli esterases. Thus, only colonies with the fastest color change should be selected. This method is not considered selection, as the cells do not have a growth advantage or disadvantage. The reaction shown here represents the staining reagent reaction. The staining solution contains a buffer to regulate the pH, and the alpha-naphthyl acetate, which is hydrolyzed by the esterase in an aqueous environment, to an alcohol, 1-naphthol, and an acid, acetic acid. 1-naphthol then reacts with the dye fast blue B, performing a violet diazonium diprecipitate. The images shown here depict the results of such a test. On the left are positive colonies with the esterase gene in the vector, while on the right are negative colonies with only the empty vector. The photos were taken 2 to 3 minutes after the start of the color reaction.